how does life do it? I mean, really, how does a single cell become a person? How does a forest grow from tiny seeds? It all comes down to one of the most incredible processes in all of nature: copying DNA. We're about to dive into the molecular machinery that makes all of life possible.
Yeah, that's the big question, isn't it? For you to grow, for cut on your skin to heal, your cells have to divide. But here's the catch. Before a cell can split in two, it has to make a perfect, complete copy of its entire instruction manual, all of its DNA. This way, each new cell gets the exact same blueprint to work from. And this is what we're talking about. On the left, you've got a single chromosome. After replication, bam, you get the structure on the right. It's now a duplicated chromosome made of two absolutely identical sister strands joined at the middle. This is step one for any cell division. But how in the world does the cell pull off such a perfect copy?
Well, to understand how you copy a blueprint, you first have to understand the blueprint itself. And let me tell you, the structure of DNA isn't just beautiful, it's absolute genius in its design for easy duplication.
So let's start with the smallest possible piece, the nucleotide. Think of it like a single letter in the genetic alphabet. Every single nucleotide has three parts. You've got a phosphate and a sugar, which link together to form the long backbone of the DNA strand. And then you have the nitrogenous base. That's the important part that actually carries the information. Now, these letters come in 4 flavors: adenine, guanine, cytosine, and thymine. We just call them A, G, C, and T. And you'll notice they're not all the same size. A and G, the purines, have this double ring structure, so they're a little bigger. C and T, the pyrimidines, are smaller, single ring guys. And this size difference, it is absolutely crucial for how they all fit together. You can just feel the excitement in that quote, can't you? When Watson and Crick figured out the double helix, they knew it was huge. Why? Because the structure itself screamed, "I can be copied." The very shape of it hinted at a simple, elegant way for life to duplicate its own code. And here is the secret sauce. It's this simple, unbreakable rule. Adenine always pairs with thymine, and cytosine always pairs with guanine. A with T, C with G. That's it. It's like a lock and key. This means if you have one strand of DNA, you automatically know the sequence of its partner. It's the key that unlocks the entire process. Okay, there's one more little detail we need, and it's a big one. The 2 DNA strands are anti-parallel. See those 5' and 3' labels? Think of them as defining the direction of traffic. The 2 strands run in opposite directions, like a 2-lane highway. This directional quirk has massive implications for how the new DNA strands actually get built. Keep that in your back pocket, because it's going to be important. All right, we've got the blueprint. Now, let's meet the construction crew. This isn't just one molecule doing all the work. It's a whole team of sophisticated enzymes, a biological assembly line, where each member has a very specific job to do. The guiding principle for this whole operation is what's called the semi-conservative model. It sounds complicated, but the idea is actually super simple and really elegant. Semi-conservative just means that each new DNA molecule that's created is half old and half new. The cell keeps one of the original strands and uses it as a perfect template to build its new partner. And you can see it right here in action. The original parent molecule unwinds, and each of its 2 strands becomes a pattern. And thanks to those base pairing rules we just talked about, A with T, C with G, the new strands that get built are exact complements of the old ones. And just like that, you get 2 identical DNA molecules where there was only one. So here's our molecular assembly line. First up, an enzyme called DNA helicase comes in and acts like a zipper, unwinding the double helix. Then, primase lays down a little RNA primer, basically saying, okay, start building right here. And then comes the star of the show, DNA polymerase. This is the master builder. It reads the template and adds the correct new letters one by one at incredible speed. Finally, after all the building is done, an enzyme called ligase comes in and acts like molecular glue, sealing up any gaps to create a perfect, continuous new strand. You know, a human chromosome can have hundreds of millions of these base pairs. Copying it from one end to the other would take forever. So, what's the cell's clever solution? It doesn't start at one end. It starts the process at hundreds of points all at once, creating these replication bubbles. The bubbles expand in both directions and eventually merge, which dramatically speeds up the whole job. It's just molecular efficiency at its absolute best. Okay, remember that anti-parallel highway we talked about?
Well, this is where things get a little weird and really clever. It turns out our master builder, DNA polymerase, can only build in one direction. It can only add new letters to the 3' end of a strand. So for one of the new strands, the leading strand, it's easy. It just builds in one long, continuous piece. But the other strand, the lagging strand, is facing the wrong way. So the cell has to build it backwards in a bunch of small chunks called Okazaki fragments, which then get stitched together. It's this brilliant little workaround to a tricky problem.
So, after all that complex, coordinated molecular chaos, what's the final result? And why is it so absolutely critical that this process is done with almost perfect precision every single time?
Well, the outcome is as simple as it is profound. You get two complete double-stranded DNA molecules that are identical to each other and identical to the parent molecule they came from. Each new cell gets one of these perfect copies, giving it all the instructions it needs to live. This is the very basis of heredity, right here.
And the accuracy is just mind-blowing. The replication machinery makes a mistake only about once in every billion letters it copies. How is that even possible? Well, DNA polymerase isn't just a builder, it's also a proofreader. It checks its own work as it goes, and if it makes a mistake, it goes back and fixes it. This incredible fidelity is what keeps our genetic code stable from one generation of cells to the next.
Think about that. This elegant, mind-bogglingly complex process is happening inside you right now, trillions of times, with near-perfect accuracy. But it's only near-perfect. And what about those incredibly rare mistakes, the ones that slip past the proofreading? Well, those are called mutations. They can be harmless, they can cause disease, or sometimes they can be the spark that drives evolution forward. So the perfect copy is what keeps us alive, but it's the occasional mistake that creates all the incredible diversity of life on Earth.